Hello and welcome to the Neuron Regeneration Podcast. This is episode 3 and the first of our news, updates, and abstract reading episodes, which are all about providing you a convenient way to stay up to date on the latest in neural regeneration. You can always access any of the papers that we discuss or any pieces of news at our show notes, where you go to neuronregeneration.com / and then the number of the episode. So in this case, neuronregeneration.com /3. And finally, for those who are interested, a lot of the abstracts that we choose to read, we're essentially looking for the keywords of neuron regeneration, neural regeneration, or nerve regeneration in a PubMed search, and then we choose an assortment of the most recently published papers. So let's dig in. All right, so here we have from the Journal of Biomedicine and Pharmacotherapy, P75 ECD-FC reverses neonatal hypoxic ischemic encephalopathy induced neurological deficits and inhibits apoptosis associated with nestin. And I'm not going to butcher the author's name. I'm just going to spell it out for you. Last name is XIAO, and first name initials are Q and X, QX. Okay, so abstract. A recent study has introduced a recombinant fusion protein consisting of the extracellular domain of P75 and the FC fragment of human immunoglobulin 1. In parentheses here, it says P75 ECD-FC, and hereafter, for simplicity, I'm just going to say P75 FC, as a multifaceted agent within the nervous system. This research aimed to assess the effects of P75 FC on neuronal growth and the restoration of neurological functions in rats afflicted with neonatal hypoxic ischemic encephalopathy, hereafter NHIE. In vitro analyses revealed that 1 micromole of P75 FC treatment markedly increased cell viability and facilitated neurite outgrowth in neurons exposed to oxygen glucose deprivation, or OGD. Subsequent in vivo studies determined that a dose of 78.6 micrograms per 3 microliters of P75FC significantly mitigated brain damage and both acute and long-term neurological impairments, outperforming the therapeutic efficacy of hypothermia, as evidenced through behavioral assessments. Additionally, in vivo immunostaining showed that P75 FC administration enhanced neuronal survival and regeneration and reduced astrocytosis and microglia activation in the cortex and hippocampus of NHIE rats. A noteworthy shift from A1 to A2 astrocyte phenotypes and from M1 to M2 microglia phenotypes was observed after P75 FC treatment. Furthermore, a co-expression of the P75 neurotrophin factor and nestin was identified, with an overexpression of nestin alleviating the neurological dysfunction induced by NHIE. Mechanistically, the neuroprotective effects of P75FC, particularly its inhibition of neuronal apoptosis post-OGD, may be attributed to nestin. Taken together, these results highlight the neuroprotective and anti-inflammatory effects of P75 FC treatment through the modulation of glial cell phenotypes and the nestin-mediated inhibition of neuronal apoptosis, positioning it as a viable therapeutic approach for NHIE. Very interesting. I'd be interested to hear where they're going to take this. Obviously, these are all recently published, and I didn't remember to say the published date. This was September 14th, 2024, which was just a day or two ago from the time of this recording. Okay, next article from the Journal of Nanobiotechnology, also on September 4th, 2024, with the author last name Yi, Y-E, first initial J. Umbilical cord blood-derived exosomes attenuate dopaminergic neuron damage of Parkinson's disease mouse model. So abstract with background. Umbilical cord blood is a rich source of multifunctional stem cells characterized by low immunogenicity. Recent research in the fields of aging and regenerative medicine has revealed that potential of human umbilical cord blood-derived exosomes, UCB exos hereafter, in promoting wound healing, anti-aging, and regeneration. However, their role in neurodegenerative diseases, specifically Parkinson's disease, or PD, remains unexplored. This study investigates the potential therapeutic effects and underlying mechanisms of UCB exos on PD. Methods: large extracellular vesicles, exos, and soluble fractions, SF, of human UCB plasma were extracted to investigate their effects on motor dysfunction of the MPTP-induced PD mouse model and identify the key components that improve PD symptoms. UCB exos were administered by the caudal vein to prevent or treat the PD mouse model. The motor function and pathological markers were detected. Differentially expressed gene and KEGG enrichment pathways were screened by transcriptome sequence. MN9D and SH-SY5Y cells were cultured and evaluated for cell viability, oxidative stress, cell cycle, and aging-related indexes by QRT-PCR, Western blot, immunofluorescence, and flow cytometry. The protein expression level of the MAPK P38 and ERK1/2 signaling pathways were detected by Western blot. Results: we observed that LEV, Exos, and SF all exhibited potential in ameliorating motor dysfunction in MPTP-induced PD mouse model, or the Parkinson's mouse model, with UCB exos demonstrating the most significant effect. UCB exos showed comparable efficacy in preventing and treating motor dysfunction, cognitive decline, and substantia nigra pathological damage in PD mice. Further investigations revealed that UCB exos could potentially alleviate oxidative damage, aging and degeneration, and energy metabolism disorders in neurons. Transcriptome sequencing results corroborated that genes differentially expressed due to UCB exos were primarily enriched in the neuroactive ligand receptor interaction, dopaminergic synapse, and the MAPK signaling pathway. We also observed that UCB exos significantly inhibited the hyperphosphorylation of the MAPK P38 and the ERK1/2 signaling pathway, both in vitro and in vivo. Interaction, dopaminergic synapse, and the MAPK signaling pathway. We also observed that UCB exos significantly inhibited the hyperphosphorylation of the MAPK P38 and the ERK1/2 signaling pathway, both in vitro and in vivo. Conclusions: our study provides a comprehensive evaluation of UCB exos on the neuroprotective effects and suggests that inhibition of hyperphosphorylation of the MAPK P38 and ERK1/2 signaling pathways by regulating transcription levels of HSB1 and PPEF2 may be the key mechanism for UCB exos to improve PD-related pathological features. My apologies for some of these things being a mouthful, even for someone who reads this stuff all the time. Okay, next paper we have from September 12th, 2024, from the Journal of Stomatology, Oral and Maxillofacial Surgery by author, last name Balaban, Balaban, first initial E. Title: The Effectiveness of Concentrated Growth Factor in Facial Nerve Crush Injury. Abstract. Aim: to evaluate the effect of concentrated growth factor on regeneration of facial nerve after crush injury. I'm going to mess up facial a bunch because I'm a myofascial physical therapist. I'm always saying fascial. Okay, so facial nerve crush injury. Materials and methods. 14 rats were randomized into two groups. The control group was an N of 7, received a crush injury of the right facial nerve. The concentrated growth factor group, I'll just say CFG group, also N of 7, received a crush injury to the right facial nerve and concentrated growth factor prepared from their own blood thereafter. Left facial nerves were used for functional comparison. Nerve function was evaluated using whisker movements and electromyography. Histological properties were evaluated using hematoxylin and eosin, and Mason trichome staining, and immunohistochemical properties were evaluated using neurofilament H and anti-tau degeneration markers. Results. In the CGF group, whisker functions began to recover earlier and recover more quickly compared with the control group. The control group showed significantly prolonged latency and reduced amplitudes in the first week compared to the CGF group. Recordings of the 4-week latency and amplitudes were similar to the preoperative period in the CGF group, whereas recordings of the same week were significantly worse in the control group. Edema and fibrosis were also more pronounced in the control group compared with the CGF group. Neurofilament H and anti-tau were at significantly high levels in the control group. Conclusion. Concentrated growth factor promotes recovery in facial crush injury and may prove a cost-effective, practical, and an effective treatment choice in peripheral nerve injury. I went ahead and clicked through. It's not a full length, but it goes into a little more depth on what exactly the concentrated growth factor is. And as a physical therapist, it's something I'm really familiar with. It's essentially a PRP or platelet-rich plasma kind of concentrate. And so let me read a little bit more about exactly what it is for those who are, whose ears perked up by the results of using this growth factor concentration. So platelets contain transforming growth factor beta 1, TGF beta 1, vascular endothelial growth factor, VEGF, insulin-like growth factor, and interleukin 1 beta. These factors improve tissue regeneration. Platelet-rich plasma concentrates were developed and evaluated for tissue regeneration. The first generation of PRPs was shown to improve the regeneration of Schwann cells. PRP has positive effects on the functional recovery of nerves, promoting enhanced stem cell growth and differentiation. Concentrated growth factor is the most recent generation of PRPs. It is produced by the centrifusing of venous blood without any additives. CGF is a gel-like substance containing platelets, leukocytes, and fibrin molecules. CGF improves the proliferation rates of bones and ligaments in the maxillofacial region. And studies using cell cultures showed increased migration of Schwann cells. However, in vivo electrophysiologic studies are required before it can be used in clinical applications. So I'd be super interested to hear from the authors what the next steps are with this particular approach and intervention. Okay, last one. I skipped a little bit down the list of most recent published articles because one caught my eye on extracorporeal shockwave therapy in spinal cord injury. And again, as a physical therapist, I've been seeing more and more about not just the results of ESWT, but anecdotally, practice owners who I work with are using it in higher and higher numbers and getting phenomenal results. Again, this is anecdotal, and this is more for musculoskeletal injury. But when I saw this in the list of very recently published, this was August 28th of 2024, I thought I definitely want to read through it and present it on this episode. So this is from the article, the Journal of Clinical Medicine. Lead author's last name, O-P-A-R-A, first initial J. So abstract. Background. Injury of the spinal cord causes motor and sensory dysfunction as well as pathologic reflexes leading to paraplegia or tetraplegia. The sequelae of traumatic spinal cord injury, hereafter SCI, are a significant burden and impact on healthcare systems. Despite constant progress in medicine, traumatic SCI still remains irreversible. To date, no satisfying treatment that can enable neuronal regeneration and recovery of function at the damaged level has been found. Hundreds of experiments have been conducted on various possibilities of influencing spinal regeneration. Some of them have yielded promising results, but unfortunately, the successes obtained in experimental animals have not translated into humans. Methods. This narrative review article presents the application of extracorporeal shockwave therapy, hereafter ESWT, in patients with SCI. The article has been divided into parts. 1. Use of ESWT for regeneration of the spinal cord after traumatic spinal cord injury. 2. Application of ESWT in spasticity after spinal cord injury. In both cases, the hypotheses of possible mechanisms of action will be described. Results and conclusions. A small number of clinical trials have demonstrated the potential of ESWT to influence the regeneration of the spine as an innovative, safe, and cost-effective treatment option for patients with SCI. Some reports have shown that ESWT can improve spasticity, walking ability, urological function, quality of life, and independence in daily life. And that's where that ends. Obviously, a little vague. I'd love to get the full length of this and get into the details of those kind of that it ended with. But I will also speak to this from my personal experience, not as a physical therapist, but rather as a father with children with spinal muscular atrophy, and looking for anything and everything that I could potentially, utilize to improve their situation, to cause neural regeneration that is at least going to be safe, if not effective. Things that are lower hanging fruit are things that we can do right away. And there's been plenty that we have found. But we looked at ESWT, and ultimately I could not get clarity on whether application of ESWT over growth plates was a precaution or a contraindication. And honestly, even if it was just a precaution, it's not something that I would just attempt on my children's spines. And for those who don't know, the growth plates in the spine don't actually fuse until you're close to 18 years old. So it's not something that, I'm guessing it's probably not in these studies been used on, kids or people with SCI younger than that for that reason. I could be wrong on that. But it was something that was like, man, I was seeing such really phenomenal results and a lot of data outside of SCI, but a lot of data that show great results in all kinds of regenerative applications with ESWT, that I was really excited to potentially be able to use it on my children, and that was kind of a no-go. So just a little additional information for those out there who are interested in this, that you do have to kind of consider growth plates in its application. Okay, we're going to wrap it up finally here with, this is actually not coming from a PubMed link, but rather from Genetic Engineering and Biotechnology News. The homepage URL there is genengnews.com. And just in September 12th, 2024, they published this article titled How the Brain Rebuilds, New Insights into Myelin Regeneration. Demyelinating diseases are any conditions that damage the protective coating on nerve cells, the myelin sheath. Some common demyelinating diseases of the central nervous system include multiple sclerosis, neuromyelitis optica spectrum disorder, transverse myelitis, and more. Now, new research led by the University of Connecticut Health and collaborators reveals a protein called C1QL1 promotes the replacement of the cells that produce myelin. The new insights may lead to the development of new and improved therapies for demyelinating diseases. The findings are published in the FEBS journal in an article titled C1QL1 Expression in Oligodendrocyte Progenitor Cells Promotes Oligodendrocyte Differentiation. And this is a quote, "Myelinating oligodendrocytes arise from the stepwise differentiation of oligodendrocyte progenitor cells, hereafter OPCs." Thank goodness I don't have to repeat that multiple times. The researchers wrote, quote, "Approximately 5% of all adult brain cells are OPCs. Why would a mature brain need such a large number of OPCs? New myelination is possibly required for higher order functions such as cognition and learning. Additionally, this pool of OPCs represents a source of new oligodendrocytes to replace those lost during injury, inflammation, or in diseases such as multiple sclerosis. How OPCs are instructed to differentiate into oligodendrocytes is poorly understood, and for reasons presently unclear, resident pools of OPCs are progressively less utilized in MS. The complement component 1, Q subcomponent-like protein family, or C1QL protein family, has been studied for their functions at neuron-to-neuron synapses, but we show that OPCs express C1QL1. We created OPC-specific conditional knockout mice and showed that C1q01 deficiency reduces the differentiation of OPCs into oligodendrocytes and reduces myelin production during both development and recovery from cuprizone-induced demyelination. That's the end of that quote. In experiments conducted in mice, the researchers observed deleting the gene that codes for COQ01 caused a delay in the rate at which oligodendrocytes mature, leading to reduced myelination of neurons. After mice were fed a drug that destroys myelin, recovery of oligodendrocytes and myelination was delayed in mice lacking the C1q01 protein. Causing mice to express more C1q01, however, led to increased numbers of oligodendrocytes and more myelination upon drug withdrawal, suggesting that C1q01 helps restore the damaged myelin layer. Thus, investigational therapies that boost C1q01 may hold promise against demyelinating diseases. Quote, our basic research on C1q01 is nascent, but there is potential that it is relevant for a novel treatment for multiple sclerosis, said corresponding author David C. Martinelli, PhD of the University of Connecticut Health Center. Quote, new drug treatment options for patients with multiple sclerosis could have a large impact on their quality of life, end quote. And that, my friend, wraps up our first episode of the news updates and abstract readings for Neuron Regeneration. I hope you enjoyed it. I hope you were able to make it through all of my verbal blunders. There's a lot of big words in there, and hopefully I'll get better, but probably not. I really do appreciate you guys being a part of the launch of this podcast. Please reach out to us and let us know if there's anything that we could do or bring to you in this show, whether it's on YouTube or through the podcast, that would be of benefit to you, that would help you move forward in your journey to help, compel breakthroughs in neuron regeneration, whether you're a researcher, an investor, a patient yourself, a caregiver, a clinician, you name it. Let us know what we can do to help accelerate breakthroughs for you and in your life, and, help do a lot of this work for you. Thank you again so much, and we'll see you in the next episode.